lecture of module 2, we will be talking about protein interactions. We had looked at the different types of structures in proteins. The aspects of protein folding mean that we have to understand now hydrogen bonding, salt bridges, van der Waals interactions and hydrophobic interactions. Now, what we mean by all these is going to give us an idea of what the protein structure in terms of how it folds into its compact three-dimensional structure is important about. So, when we look at the protein architecture, we found out that the primary structure was just a string of amino acids that were connected together by the peptide bonds. For the secondary structures, when we look at these 4 levels, for the secondary structure what we had is we have the alpha helices and the beta sheets. The different types of alpha helices were the regular right-handed alpha helix, then we had the 310 helix and the pi helix. For the beta sheets, we had the antiparallel beta sheets, and we found out the different levels of hydrogen bonding in terms of the secondary structural elements of the protein as they are called. We then went on to look at the tertiary structure of the protein that formed the final native structure of the protein, in the sense that we have the different interactions that we are going to look at in this lecture to study how they come together. And in the quaternary structure, we learned that there are different subunits of some proteins that come together to form a fully functional unit. Now, what these proteins are is the single polypeptide chains that have these amino acids in a specific sequence that interact with each other in a three-dimensional arrangement. So, the fact that this chain is coming together to form this compact three-dimensional structure is what is extremely important, because the way it forms its three-dimensional structure is important, because we are going to have amino acids at different parts of the protein come together to form specific geometry, which is important for the active site or for other interactions of the protein. So, when we talk about the weak interactions, they are important in the folding of the polypeptide chains into their secondary and tertiary structures. So, what are these? These are the weak non-covalent interactions. We know that the connection is through the peptide bond, and the other covalent bond that we have is the disulfide linkage. So, we have hydrogen bonding, hydrophobic interactions, ionic interactions, van der Waals interactions. And of course, we have the disulfide linkages, as I mentioned, that bring the different parts of the protein together in the formation of another type of covalent bond that is present. So, when we look at these different types of the different structures that are possible, our idea is now to see how we can understand. So, the disulfide bond we know is a formation of the cysteine-cysteine residues that form the cysteine bond. We have hydrophobic interactions, we have hydrogen bonds. Hydrogen bonds we have seen between specific residues in the alpha helix, specific strands for the parallel and the antiparallel beta strands. Now, what we are going to look at is we are going to look at the specific types that bring about what is called protein folding. Now, what is protein folding? So, that we have this long chain of the amino acids that are linked to each other. We know that we have them linked by the peptide bonds. Now, this forms this three-dimensional structure. So, there is a process involved, and this process is called protein folding. And what is this process? It is the process by which the long, flexible, unbranched chain of amino acids, that is the unfolded form, will spontaneously form a stable three-dimensional structure that we refer to as the folded form or the native form of the protein. So, this is important because we need, for this protein to function, it has to fold in this fashion. So, we are going to look into the process of protein folding, but before we do that, we have to look at the specific interactions. So, the specific folding or the specific interactions that we observe are important because the process important. Why? Because the folding will ultimately decide the biological function of the protein. So, the dominant contributors to protein folding are the hydrophobic effect, hydrogen bonding, Coulombic interactions and van der Waals interactions. So, we finally get folded structures that look like this. Now, hydrogen bonding. We looked at hydrogen bonding when we considered the peptide bond. Now, what we are saying is we know that we have the backbone. The protein backbone contains the C double bond O and the NH. Every amino acid is interacting with the next amino acid through a peptide bond. And what is this peptide bond? It is the C double bond O NH. So when we have specific interactions like this between the C double bond O that we see here and the NH that we see here, we have a specific hydrogen bond formation. A hydrogen bond formation we know is between electronegative atoms where we have a hydrogen connected to one of them. There is a strong interaction between the electronegative atom of one and the hydrogen of the other, giving rise to hydrogen bonds. Now, these hydrogen bonds are important in proteins, not only in the backbone, but also in the side chains. As we looked at, there are specific polar amino acid side chains that are involved in hydrogen bonding. So, a knowledge of the amino acids is important to understand which one of these can actually be involved in hydrogen bonding. So, when we look at the hydrogen bonding network, say for a protein like lysozyme, we will see that this network involves not only the backbone atoms, but also side chain interactions are possible. So, if we just look at, so this would be a side chain interaction where we are looking at contributions not only within the protein itself, but also with the surrounding water molecules that can be involved in a hydrogen bonding network with the proteins. So, this important network is essential for the protein to remain stable in its three-dimensional framework. The salt bridge, as the name implies, are formed between oppositely charged groups by electrostatic interactions, and we will look at the relative strength of each of these types of interactions as we go on in the course in looking at protein stability, which will be part of our next module. Now, when we look at salt bridges, we then look at a positive charge and a negative charge. So, in this case we have a protein that has an arginine residue and a retinoic acid, which is a ligand, and we see the possibility of a formation of a salt bridge that brings about a stability. So, this is important in understanding how even amino acids of different charge can come together in an electrostatic interaction, electrostatic attraction that is going to bring different parts of the molecule together, or also interact in a manner that is going to be important for the protein to maintain its three-dimensional structure. So, when we look at an aspartic acid residue, or for that matter for any such residue, the example of aspartic acid and arginine is given at the top here. We can also have an NH3 plus and a COO minus. So, when we have an NH3 plus and a COO minus, again we have the possibility of these ionic bond formation, because we have opposite charges that are going to interact with each other. So, when we have these interactions, the favorable interactions between the opposite charges, and another example would be looking at lysine. So, when we have a lysine example, we know that we have, this is the, so when we have these lines, these wiggly lines, they usually mean that the rest of the protein follows here. And what we have here is, we know that this is the CO and the NH. So, this is where we have the connectivity, the peptide bond. So, this is a lysine residue, and we can have it connect to a glutamate acid residue. How do we know this is glutamate? We know that there is a CH2 here and a CH2 here. So, this is a glutamic acid. So, we have to know how to identify the amino acids, and then also know from a knowledge of the type of interactions or the type of side chain that they are involved in, what kind of interactions are possible between the specific side chains. So, when we are looking at the side chain of lysine and glutamic acid, we see that there is a possibility of an electrostatic interaction because of the charges, the negative charge and the positive charge. In addition to that, we can also have hydrogen bonding possibilities. So, what is happening here is we have a mode by which we can have a strong interaction, and this helps in the stability of the protein in its overall native conformation. The other information that we need to know is van der Waals interactions. Now, in van der Waals interactions, we know that every atom has an electron cloud around it. Now, this electron cloud is constantly fluctuating, and because of this there is a temporary electric dipole that is formed. Now, because of this temporary dipole, there can be favored interactions between the atoms. They are weak transient electrical attractions from one atom to the other that give rise to what are called these van der Waals interactions, that even though weak in nature, they can accumulate because there can be a large number of such van der Waals interactions in the protein. In the hydrophobic interactions, what we see, this is the tendency or the preference for the non-polar surfaces to remain in contact with each other rather than the polar solvent water. So, in this case what happens, if we have specific side chains that do not have an oxygen and a nitrogen associated with these. Now, we have to remember, when we are talking about the side chains, in this case we are talking about the additional attachments to the C alpha, because each amino acid is always going to have its N terminus and its C terminus and the hydrogen, and to this is going to be attached an R group. Now, this R group is what we are interested in. So, if this R group happens to have carbon and hydrogen in it, then what is going to happen is the interactions between them, no electrostatic interactions are possible. No hydrogen bonding is possible because the side chain does not have an oxygen or a nitrogen. So, what happens is we have these hydrophobic residues that, interestingly for the globular proteins, as would be expected, would tend to remain away from the surface of the protein. So, what happens is, when we look at a regular globular protein and we look at all the hydrophobic residues, so we have to identify which are the hydrophobic residues, and that is something we have to know. Which ones are the side chains that have carbon and hydrogen in their present in their R group. So, these are the ones that are going to be hydrophobic in nature, and they would preferably interact with each other rather than with the solvent. So, then what would happen is they would tend to remain away from the surface, and what we would see is we would see them bunching up together or forming what is known as the hydrophobic core of the protein when it folds in this fashion in a regular globular protein. So, when we look at these hydrophobic interactions, we realize that because of the specific side chains that are involved, they are going to interact in a manner that is going to form the interior hydrophobic protein core, where what happens, where most of the hydrophobic side chains, they remain closely associated with each other to be shielded from interactions with the solvent. And with the knowledge of the different types of amino acid residues, looking at which side chains can be involved in what type of interactions, that is extremely important. So, with the knowledge that we have the basic amino acid residues, we have the acidic amino acid residues that can be involved in ionic or electrostatic interactions. When we look at the polar type that have an oxygen and nitrogen to it, but those would be preferably be involved in hydrogen bonding type interactions, and the hydrophobic ones would be the ones that have the C and the H in them. So, these would preferably remain in the hydrophobic core of the protein. So, when we have these polypeptide subunits in the quaternary structures, they are held together by the hydrophobic interactions, hydrogen bonding and ion pairs. So, what are we now looking at? We are looking at the, now that we have the folded compact structure and in a regular globular protein, we know that we have the hydrophobic core and the rest of the amino acids on the surface. Now, what happens if we have a dimeric protein? So, I have two tertiary structural units, both in their subunit form, two monomeric units come to form a dimer. So, the question is, what is connecting these two together and why do they remain as a unit like this? So, that is important. What are the interactions that are involved in the subunits coming together? So, for example, when we look at hemoglobin, we have a subunit composition as we saw in the last lecture, alpha 2, beta 2. So, why is it not that the alpha and the beta are disintegrating? So, what is holding these together? This is also important. So, we have a dimer, an oligomer of two protomers. These are alpha B protomers in the sense that we have the alpha 2 and the beta 2, right. So, now when we have the association, so in the if we have a single unit that is like this and we have another unit that is like this, there is a certain terminology that we use called the surface area. So, if this is the surface area of the protein or of that specific subunit that is lost when it interacts with the surface of another subunit, regularly in the normal case, this part would be exposed to the solvent. So, would the darkened part on the other chain. So, say we are looking at say an alpha and a beta. So, the alpha chain part would have been exposed, the beta chain part would have also been exposed, but when they come together, the association can typically what is called bury around 1000 to 2000 angstroms of surface area that would have been otherwise exposed to solvent. Now, the fact that we have these two units coming together means there are specific interactions that are going to hold these together in a specific formation like this, and what is observed in the oligomeric units formation, we preferably see hydrophobic interactions to a greater extent in these types of interactions. So, an average of 77% of inter-subunit hydrogen bonds are between the side chains. We have to hold the subunits together, and the hydrogen bonds generally within the subunits are between the backbone atoms, and the secondary structural elements that we observe are not generally carried over the subunit boundaries. So, these would be two subunits that would be held together in a fashion that could include hydrogen bonds in addition to preferable hydrophobic interactions. So, now that we have a knowledge of a three-dimensional structure of a protein, an idea of what the forces that are holding these together could imply, we want to look at another aspect of a protein structural sense in understanding what a contact map is. Now, to have a contact map of a protein, what we need to know here is we need to know what are called the coordinates. So, in the coordinate, what do we mean by coordinates? When we have a structure, for example, if we look at the specific structure here, we know that we have specific side chain atoms. Now, each of these side chain atoms here that are drawn in circles here, spheres here, they have a position in three-dimensional space. So, if we can look at the three-dimensional position of each of these atoms for the specific side chains or for the specific amino acids, we can construct what is called a contact map. Now, the reason we try to look at a contact map is apparent later when we try and understand structural aspects, but when we are looking at a specific, so if I look at a specific amino acid and I just look at the C alpha of this specific amino acid and I try to find out the distance between another C alpha from somewhere far in the protein, there is a specific distance d that I am looking at. So, the idea in the construction of the contact map is to look at all C alpha of I, connect all C alpha of j and find the distances. Now, this is, so we have say AI and AJ, these are two different amino acids that are connected in contact if their 3D distance is less than a contact threshold. So, what would that mean? That means, if I consider C alpha of residue 1 to be close to C alpha of residue 2, this is what I would expect, because they are in the sequence one after the other. So, we are looking at interactions, or we are looking at specific ways in which we could look at distances between the C alpha carbon atoms in an identification of a structural aspects in what we call a contact map. So, the contact map will be described in a later slide. So, what we have is we have a matrix representation. So, where do we get this matrix representation from? We have a binary two-dimensional matrix that does what? It shows the distance between all possible amino acid residue pairs of a three-dimensional protein structure. So, what this means is that we have the C alpha of residue I and we have the C alpha of residue j. What do we have of the C alpha of residue I? We have the x, y, z coordinates in three-dimensional space. For C alpha j, we also have the three-dimensional coordinates in space, and what we can calculate is we can calculate the distance from C alpha I to C alpha j. Now, if we were to do this exercise from every atom to every other atom along the two, so this would be say from residue 1 to residue, BSA is a very large protein, say close to around 600 residues. So, this would mean that we have a contact map between the C alpha of 1 to C alpha of 2, C alpha of 1 to C alpha of 3, and we create what we call this two-dimensional binary matrix. Now, what is the information that we can get from this matrix? This is important. Interestingly, we can look at secondary structures and the contact map. And what we get from these secondary structures is we have alpha helices that appear as thick bands along the main diagonal because they involve contacts between one amino acid and its 4 successors. What do we mean by that? We mean that we have a hydrogen bonding we know from I to I plus 4. Now, because we have the compact structure in the alpha helix, it would mean that if residue number say 3 to 12 were to form an alpha helix, what would I expect? This is the question that I am asking. So, this means that 3 is close to 4, it is close to 5, it is close to 6, it is close to 7, and there is a hydrogen bonding between 3 and 7. Meaning that 3 and 7 are going to be close to in three-dimensional space. Also, there are signatures of parallel or anti-parallel beta sheets that are thin bands that are parallel or anti-parallel to the main diagonal. So, essentially what we are doing is for a protein of n residues, it means we would have an n into n matrix, and the elements would tell us whether the two parts of the protein or the two residues that we are looking at are close enough together or not. Let us look at what this means. So, essentially what we are doing is we are taking the C alpha of I and we are calculating the distance to C alpha of j. What we do is we create a matrix. We create a matrix and we say that if we have say 60 residues, so I look at 1 to 60 here, and I also look at 1 to 60 in my along the y axis. What we have in this case is all along the diagonal, what I would expect is I would expect a thick line, because 1 is close to 2, which is close to 3, 2 is close to 3, 3 is close to 4, and so on and so forth. So, I would expect a line, and we also have to know that the upper and the lower triangle of this matrix is going to be the same. because if residue number 3 is in contact with 7, then obviously 7 is in contact with 3. So, now if I have say a helix, say I have a helix around this region. Now, what do we know is we know that helix are contiguous in nature. So, say I have a helix from around say 25 to 35. So, what would I expect? I would expect 25 to be close to 26, which is natural, but I also expect a hydrogen bonding between I and I plus 4, which means there is going to be a thicker band along this helix region telling me that residue number say 25 is in contact at 29. So, a thick region along the diagonal would imply that there is an alpha helix, because they are close together in 3-dimensional space and we have a specific hydrogen bonding pattern that is bringing them close to each other. When we are looking at the parallel and the anti-parallel beta sheets, let us see what we see now. Now, say I have a parallel beta sheet where of the strands of which are say from 2 to 8, and again I have, if they are anti-parallel, so 2 to 8 will be in this direction and say 44 to 52 would be in this direction. So, this is an anti-parallel. Now, we know we have hydrogen bonding patterns between this. So, say I say 8 is close to 44. So, the way I would plot this, I would have 8 say somewhere around here, that is close to say 44. Then I would have 7 that is close to 45. So, then I would have, so if this is 8, so let us just mark it here. So, this would be 44 and this would be 8. Now, I say 7 because I am 1 back here and 45. So, I am 7 here and 45 would be up here. So, I would have this here. Then if I look at number 6, that would be in connection with 46. So, I go back a step here, further up here. What would I see? I would see a line that looks like this. So, this would be an anti-parallel beta sheet. Now, say I have a parallel strand. So, I have a parallel strand that takes me say from let us go to a region we have not considered, say from 10 to 17, and I have a parallel strand that is from 34 to say 40. So, now when I am looking at hydrogen bonding patterns here, I would have 10 close to 34. So, 10 would be here and 34 say somewhere here. Then we have 11 close to 35. So, I would look at 1 here and 35, which would be another point here. Then I would look at 12 and I would look at say 36. So, then I would be again 1 on the right, and so what I see is I see that I have a parallel connectivity. So, when I look at specific regions of the protein in a contact map from the 3-dimensional coordinates, that is the x, y, z information that I have, I can identify specific secondary structures on the contact map. For example, if I have now an extra dot here, this extra dot may give me some information saying that there is 58 and say, if I look at the point here, say this is around 40. So, I would say 40 and 58 are close together in 3-dimensional space. Now, the question may arise as to why are these two particular residues close to one another. It could be that it forms a disulfide bond. It could be that there is an electrostatic interaction. So, from a knowledge of the contact map, it is easy to identify specific not only secondary structures in the contact map, but also information about which parts of the protein are close to one another. And this is particularly important when considering protein dynamics, as to understand which regions are changing in three-dimensional space when we are allowing a changing of the coordinates due to interactions or due to ligand binding. So, if we look at the contact map for a specific protein here, we see that if we look at these two strands now, this we can see is, so here is the N terminus. Why do we know this is the N terminus? Because this is where the protein, we cannot see, but we can see that the arrow ends here. So, this has to be the C terminus of the protein. So, we have what we have here, then we have the alpha helix, and then let us try and trace this. So, here is where it begins, then we go down this way around this to form this beta strand. Then we have the formation of the helix, and then we have this beta strand that then forms the other beta strand. So, what we do have is we have two beta strands in so this is an antiparallel beta strand, here also we have an antiparallel beta strand, and we have an helix. So, now if we look, now we understand what amino acid AI and what amino acid AJ means. That means, and what are these points on the graph, or what are these red boxes that we have here? The red boxes are indicating that these residues that are located here are close to each other in three-dimensional space. So, if we try and now identify the regions of the protein that are close to one another in the specific types of beta sheets or the beta strands that we look at, and the alpha helix that we can see in the. So, what we have is, if we look at this particular one, this one that starts and the one that ends, these strands are in the same direction, giving us a parallel set, right. And what we have in the other cases here, for this particular beta, these two beta strands are antiparallel, and so are these two beta strands that are antiparallel, and the one in these two in the middle are parallel. So, this is very clear from the structure of the protein in three-dimensional space. And we also see the helix, and what we did learn for the helix was, along the diagonal we are going to have an extended portion. So, this portion will correspond to the alpha helix. Why? Because we know that we are going to have this close in three-dimensional space, because there is specific hydrogen bonding for a regular alpha helix, say between I to I plus 4. So, what we have here is, we have the parallel beta sheets, as we saw in the previous slide as I explained it to you. These are the antiparallel beta sheets, and these are the, or rather this is the alpha helix. So, we have now, this is another antiparallel beta sheet, and so we have one antiparallel beta sheet here, one antiparallel beta sheet here, a parallel beta sheet shown here, and the alpha helix. So, now say we did not have the structure on the right, from the information present here we are going from residue number 1 to residue number 60. We can identify where we have the alpha helix in the protein, where we have the antiparallel beta sheets in the protein, and where we have the parallel beta sheets in the protein. In addition to that, we have additional information about which parts of the protein are close to each other in three-dimensional space. So, the information that we can get from the contact map is interesting, giving us an idea about how we can plot the information, given that we know the structure. What is the structure? The three-dimensional coordinates of all the atoms present, but what we plot here for convenience is the C alpha coordinates, and we have the specific distances associated with it. So, what we looked at is we looked at general protein folding, the process that brings the amino acids that are linked in the primary structure to form the secondary structure, then the tertiary structure forming the native three-dimensional unit of the protein. We have the hydrogen bonding that we looked at, and what we realized is we need to know which amino acid residues can actually be involved in the hydrogen bonding that is present, the salt bridges where we would look at electrostatic interactions and what kinds of residues again could be involved in these salt bridges, van der Waals interactions and hydrophobic interactions. So, these are the concepts that we looked at. We will look at further interactions and a further understanding in our next lecture. Thank you.